Good afternoon, everyone. My name is Brandon Monstein. I am a junior at Florida International University, and I'm excited to present a fascinating study published in 2023 by Kosia and colleagues in the Journal of Cell Science. This study examines the role of myo-19, a myosin motor protein, in mitochondrial fission. Unlike most myosins that transport cargo along actin filaments, myo-19 appears to function in a different way, as a tether that physically connects mitochondria to actin filaments associated with the endoplasmic reticulum, or ER. Throughout this presentation, I'll walk you through the background, methods, key findings, and broader implications of this research and how it reshapes our understandings of how cells regulate mitochondrial dynamics. Goal of the study. The primary goal of this study was to investigate whether myo-19 plays a functional role in promoting mitochondrial fission by tethering mitochondria to ER-associated actin filaments. The researchers hypothesized that rather than acting as a conventional transport motor, myo-19 facilitates mitochondrial fragmentation by holding the organelle in place at sites where fission occurs. Specifically, they asked whether myo-19's ability to bind actin without necessarily generating motor force was enough to promote mitochondrial division. They also aim to clarify how myo-19 interacts with other cellular components, such as ER-anchored formins and mitochondrial outer membrane proteins, to position mitochondria for fission. This goal stems from the need to better understand upstream events that lead to the recruitment of the fission protein, DRP1, especially at ER mitochondria contact sites. Background. To fully appreciate the role of myo-19, it's important to understand the biological context of mitochondrial fission. Mitochondria are not static. They constantly undergo fission and fusion to maintain their function and morphology. Fission is essential for several cellular processes, including mitophagy, energy production, and inheritance of mitochondria during cell division. This regulation of fission is linked to neurodegenerative diseases and metabolic disorders. The fission process often begins at contact sites between the mitochondria and the ER. At these contact points, actin filaments are polymerized with the help of proteins such as Spire1C and INF2 CAAX. These actin structures serve as scaffolds for the recruitment of DRP1, a GTPase that ultimately constricts and severs the mitochondrion. The precise coordination of actin polymerization, organelle tethering, and protein recruitment is essential for proper mitochondrial fission. Introducing myo-19. Myo-19 is a relatively unique member of the myosin superfamily. While many myosins function to transport cargo along actin filaments, myo-19 has been found to localize to the outer mitochondrial membrane. Its tail domain interacts with proteins like myro1 and myro2, which anchor it to the mitochondria. Myo-19 is known to have a high duty ratio, meaning it stays bound to actin for a longer portion of its mechanochemical cycle. Excuse me. This property makes it ideal for functions involving anchoring or tethering rather than movement. Previous research has implicated myo-19 in mitochondrial motility, especially in structures like philopodia and in organizing mitochondria during cell division. However, its potential role in promoting mitochondrial fission through stable actin binding rather than cargo transport had not been explored in detail until this very study. Now, on to the summary of the techniques or the experimental methods. To test their hypothesis, the researchers employed a comprehensive set of experimental techniques. First, they used siRNA to knock down myo-19 and various related proteins, including INF2 CAAX, Spire1C, and the metaxins MTX1 and MTX3. They also used plasmids to overexpress both wild type and mutant forms of MYO19. Imaging was a major component of the study. They used confocal microscopy to observe mitochondrial morphology, STORM, stochastic optical reconstruction microscopy, to assess MYO19 distribution on mitochondria, and TIRF, total internal reflection fluorescence microscopy, to measure photobleaching and determine molecular stoichiometry. Additionally, they use split luciferase reporter system to measure ER mitochondrial contact proximity. This broad toolkit allowed the team to assess both the physical and functional aspects of MYO19's role in mitochondrial fission. MYO19 knockdown effects. One of the first experiments in this study involved knocking down MYO19 using siRNA. The results were striking. In the absence of MYO19, the mitochondrial network became highly elongated and interconnected, a phenotype commonly associated with reduced fission or excessive fusion. This phenotype was consistent across both HeLa and C2C12 cells, suggesting a generalizable role for MYO19. The knockdown efficiency was confirmed via Western blot, which showed over 96% depletion of MYO19 protein, especially these hyperfused mitochondria did not cluster near their nucleus, indicating the general mitochondrial motility wasn't impaired, just the fission process itself. These findings strongly suggested that MYO19 is necessary for maintaining normal mitochondrial morphology and may act as a positive regulator of fission. Overexpression effects. After showing that MYO19 depletion led to elongated mitochondria, the researchers explored the effects of overexpressing MYO19. When wild-type MYO19 was overexpressed, mitochondria became fragmented, essentially the inverse of what was observed in a knockdown condition. This effect was consistent whether the MYO19 was tagged on the N-terminus or C-terminus, ruling out any artifact from the tagging. The consistency of these results across different cell types and expression systems emphasized that the phenotype was truly driven by MYO19 activity. This dose-dependent relationship further supported MYO19's role as a mediator of mitochondrial fission. Notably, this overexpression phenotype mimicked those seen with actin overproducing constructs, indicating that MYO19's role may involve interacting with or tethering to the actin cytoskeleton rather than moving along it. Actin binding versus motor force. The researchers next aimed to figure out which part of MYO19 function was crucial for promoting mitochondrial fragmentation, its ability to bind actin or its motor force generation. They created specific mutants of MYO19, one that weakened actin binding, which was G417A, and others that disrupted the motor stroke, Delta 2, 1Q, and R659L. The results were very clear. Cells expressing the G417A mutant showed no mitochondrial fragmentation, meaning the actin binding was absolutely necessary. However, the motor mutants, despite being defective in movement, still caused fragmentation. This demonstrated that MYO19's force generating ability was not essential. Rather, its tethering, simply binding to actin and holding mitochondria in place, was a critical function. Localization and structure of MYO19. Using STORM super resolution microscopy, the authors studied how MYO19 was distributed across the mitochondrial surface. They found that MYO19 molecules were sparsely and evenly distributed, with about 130 nanometers between them. Photobleaching experiments confirmed that most MYO19 molecules exist as single units, monomers, instead of forming clusters or multimers. This sparse distribution and monomeric form are consistent with a tethering function rather than a transport function, which would typically require clustering or cooperative movement. This structural arrangement supports the idea that MYO19 acts as a small strategic anchor on the mitochondrial surface. Metaxans anchor MYO19. The study also investigated how MYO19 is localized to mitochondria. The researchers found that two mitochondrial outer membrane proteins, MTX1 and MTX3, were essential for MYO19 anchoring. Knockdown of both resulted in significant decrease in mitochondrial MYO19 levels. Depleting both did not significantly affect MYO19 localization. This suggests that the metataxins are a major platform for stabilizing MYO19 on a mitochondria, allowing it to bind to nearby actin filaments and perform its tethering role. Actin source for MYO19. Give me 1 second. Excuse me. The next question was, where does the actin that MYO19 binds to come from? To answer this question, the researchers looked at two key actin nucleating proteins, Spire1C and INF2-CAAX. Both are involved in polymerizing actin filaments at ER mitochondrial sites. When neither Spire1C or INF2-CAAX was knocked down, MYO19 could no longer promote mitochondrial fragmentation. This indicates that MYO19 needs the actin filaments gathered by these two nucleators. Mitochondria contact sites. To directly measure mitochondria contact sites, the researcher used a split luciferase proximity assay. In this system, luciferase fragments are targeted to the and mitochondria. An luminescent signal appears only when the two organelles are in close proximity. MYO19 knockdown caused a 40% drop in this luminescence signal, confirming that mitochondria contact were significantly reduced. One major finding was that even though MYO19 can tether mitochondria to actin and stabilize contact, it cannot complete mitochondrial division on its own. It still requires the action of DRP1. When the researchers co-expressed a dominant negative version of DRP1, MYO19-driven fragmentation was blocked. This showed that MYO19 acts upstream of DRP1, creating the spatial and temporal conditions needed for DRP1 to be recruited and to drive the final membrane constrictions and scission. Now on to the approach. Combining all the results, Combining all the results, the researchers proposed a clear model. MYO19 acts as a static tether that binds mitochondria to associated actin filaments at fission sites. This study not only sheds light on mitochondrial dynamics, but also changes how we think about myosin proteins. Traditionally viewed as movers, MYO19 demonstrates that myosins can also act as stabilizers, ensuring spatial positioning rather than cargo transport. This highlights the growing recognition of intracellular architecture as a dynamic and actively maintained system, not just a passive scaffold. MYO19's role in stabilizing mitochondria contacts has potential implications for diseases involving mitochondrial dysfunction, such as neurodegeneration and metabolic syndromes. Broader implications. Beyond its immediate finding, this study opens the door to broader questions about cell biology. Are there any other tethered myosins anchoring different organelles? Can MYO19 also regulate calcium transfer between and mitochondria, influencing bioenergetics and signaling pathways? Future studies could explore MYO19's role under cellular stress conditions, such as oxidative damage or nutrient deprivation, where mitochondria often fragment in response to changing metabolic needs. Conclusion. In conclusion, this research showed that MYO19 is essential for mitochondrial fission, but not by moving mitochondria, but by anchoring them. It stabilizes mitochondria contact points, allowing actin filaments and DRP1 to cooperate in completing fission. This study not only clarifies a crucial step in mitochondrial division, but also broadens our understanding of how cells organize their internal architecture. MYO19's tethering function represents a new but exciting layer of cellular control over organelle dynamics. Thank you so much for your time and attention. This study provides an excellent example of how even well-known families of proteins like myosins can surprise us with unexpected functions. If you have any further questions, just reach out to me. Again, my name is Brandon Monstein, a junior at Florida International University. This is a presentation for my cell biology class. Thank you for anyone who watched this presentation, and thank you.